This is the lecture for Chapter 6, Metabolism, Energy and Enzymes. In a previous lecture, I talked about all the different characteristics of life, and these are characteristics that all living things share. And with these characteristics, a lot of them relate to energy. And one involves energy processing. This is metabolism, so that directly relates to energy. Then there's regulation, homeostasis, maintaining a constant environment. And then other characteristics that require energy would be growth and development, reproduction, and responding to the environment. And even order, having a particular order in a multicellular organism or a cell, having a particular order with its organelles, that also requires energy. Metabolism is one of those characteristics of living things or characteristics of life, and metabolism is required for all living things, whether they are a multicellular organism like a human or a unicellular organism like an individual cell. Either way, all living things must take in food, they must take in nutrients because they're going to take those nutrients, take that food in, break it down into its subunits, the individual polymers, and then use those polymers to build specific molecules that the cell needs. So cells do that and multicellular organisms do that on a larger basis. Either way, if it's just an individual cell or a multicellular organism, all of this metabolism requires energy. So living things require a source of energy. When talking about energy, it's important to define what energy is and talk about different types of energy. So the basic definition for energy is the ability to do work. And there are two overall types of energy: potential energy and kinetic energy. And potential energy, this is basically defined as stored energy. So it has the capacity to do work. That's potential energy. And so this ball that is held over the ground, held up, is going to have potential energy. Kinetic energy is the energy of motion. And so this ball, once it is dropped, then it has kinetic energy. As it is moving down, falling down, that's energy of motion. Now there are a couple different types of energy. First is mechanical energy. And mechanical energy involves kinetic energy. And mechanical energy is what you study in physics or engineering. So these are structures that when you build them, they have the potential to do work, and then as you are using them, they have kinetic energy, so energy of motion. So some examples of this are the pulley, the wedge, the screw, the lever, the inclined plane, and the wheel and axle. So those are examples of mechanical energy. And you can have potential mechanical energy and kinetic mechanical energy. In addition to mechanical energy, there is chemical energy. And chemical energy is the energy of molecules. So this is what you often study in chemistry. So when you look at the different molecules that you talk about in chemistry, they all have a certain level of potential energy, and then when they're involved in a reaction, they will release that energy, and then you often have kinetic energy. And as you look at this, a lot of this involves combustion, burning. So you have a reaction of potassium with water, which creates flames. You have a burning matchstick, burning wood, burning candle, burning natural gas. Batteries are a different type of chemical energy. This is generating an electrical current. So it's not burning, but it's generating an electrical current. And then you have respiration. And here respiration doesn't mean breathing, it means metabolism. So we call that cellular respiration. In this class, of course, we're going to focus on energy that relates to living things. And all living things, like I said before, have to take in food. They have to take in nutrients. So food basically is made of molecules, and this is potential energy for the cell. So it has stored energy that the cells are going to be able to use and convert into kinetic energy, usable energy. The laws of thermodynamics relate to energy, and you often study these in the areas of physics, chemistry, and engineering, but they do apply to living things also. And two of the laws I want you to know for biology. The first one is the first law of thermodynamics, and that is called the law of conservation of energy. And basically this means that energy cannot be created or destroyed. So you must have energy present before, and then you can convert it into another type of energy, but in doing so, you're not going to destroy any of that previous energy. So this applies to living cells because cells need energy. So they must get energy from some place, and they can often convert that energy into another form. But in doing so, the cell cannot create its own energy. That goes against the laws of thermodynamics. And when it uses the energy, it's not going to destroy the energy, it just changes its form. So first law of thermodynamics, the law of conservation of energy, cells have to follow that law. And then the second law, it relates to entropy. And entropy is basically disorder. So it's disorder. And this is the idea where things that are organized in the universe, they are moving towards disorder. So that's sort of the natural movement of things. So like a highly organized molecule made of hundreds of thousands of different atoms, eventually it's going to break down into those different atoms. So things in general tend to move from an ordered organization to disorder. And this relates to the first law of thermodynamics, because as you take energy and convert it to another form of energy, you're going to lose some of that energy to entropy. And often this is associated with heat. So for living things, as they convert energy from one form to another, they lose some of that energy in the form of heat. And heat is a very disordered form of energy. The laws of thermodynamics and the different types of energy I have talked about already can be used to explain the flow of energy through an ecosystem. So this is an example of a terrestrial ecosystem, and it shows how energy moves through the plants and animals. And this is also true with aquatic ecosystems also. So first, law of thermodynamics, the first law, energy cannot be created or destroyed. So the original source of the energy that moves through the ecosystem is the sun. So that energy then interacts with the plants, and the plants convert that energy into chemical energy into the form of glucose. So solar energy is converted into glucose, and then the moose comes along, eats the glucose, and converts that chemical energy into ATP, another form of chemical energy. In terms of potential and kinetic energy, the energy that comes from the sun, originally it's in a kinetic form because those rays are moving through outer space, moving through the sky. They encounter the plant converts that into potential energy. So glucose has potential energy, and then when the moose eats the glucose and metabolizes the glucose, it can convert that potential energy of the food into mechanical energy so it can move. And with each energy conversion, you are losing some of that energy as heat. So for every conversion, changing energy from one form to another, you're losing some as heat. And in general, you lose about 50% of the energy every time you convert it. On a cellular level, there is also a flow of energy. So here is the light energy from the sun. Photosynthesis is the process which converts that light energy into chemical energy, potential chemical energy. Then cellular respiration takes that glucose and converts it into ATP. And ATP is the readily usable form of energy that the cell uses to perform most of its work, so any sort of kinetic work that it needs. And During all those processes, a lot of the energy is released as heat. So anytime you convert it from one form to another, you're going to lose a lot of that energy in the form of heat, which relates to entropy, increasing disorder in the universe. Now I want to focus on how energy is involved with living things. And when we talk about that, we often refer to metabolism. And metabolism is defined as the sum of all chemical reactions that occur in a living thing. And this can either be on an individual cellular level or it can be on an organismal level. So that would apply to multicellular organisms. But basically, metabolism takes energy. So again, metabolism is going through taking in nutrients, catalyzing certain types of reactions to produce certain products, and all of that requires energy, and it involves producing heat. So remember, heat is that disordered form of energy, increasing entropy in the environment. Chemical reactions are what you study in chemistry, but they are also important for biology. So when talking about metabolism or photosynthesis or cellular respiration, we're going to be going over some reactions. So it's important to know the basics of chemical reactions. When looking at a chemical reaction, on the left side, you write down all the reactants, and then on the left side of the reaction, you have the products. And the arrow shows the direction of the reaction. So this is a very simple reaction of how to form water. If you want to form water, you need to have hydrogen and oxygen as your reactants. And then the reaction, if it goes forward, you will form water molecules as the products. Another example is how to form ammonia. If you want to form ammonia, you need nitrogen atoms, hydrogen atoms as your reactants, and then the ammonia itself is the product. Also, when talking about reactions, you have to have an idea of how energy is involved. Basically, reactions can be either exergonic or endergonic, and this has to do with the net release of energy. So in an exergonic reaction, the reactants have more potential energy, higher potential energy than the products. So when the reaction goes forward, the products end up with less energy. So that means there was a net release of energy. And this usually indicates that the reaction was spontaneous. Endergonic reactions are the opposite. So in an endergonic reaction, the reactants have less energy than the products. So when the reaction goes forward, in order to get that reaction to go forward, you have a net input of energy. So it requires more energy to complete that reaction than you get out of it. And this reaction is non-spontaneous. So that's not going to spontaneously happen. And often in biology, in the metabolism of a cell, exergonic reactions are coupled with endergonic reactions. So often the energy that is released from the exergonic reaction is put into the endergonic reaction. That's where the energy comes to put into that reaction. Biochemical reactions are chemical reactions that relate to living things, so biology, life. And when talking about biochemical reactions, there are three important aspects that you need to know about them. First, what is happening on a molecular level? Are you making large molecules, or are you taking large molecules and breaking them down? Second thing to keep in mind is how water is involved. Water is very important for living things. It's probably where life evolved, and it's a very important part of biochemistry. If you don't have water, the cells won't be able to metabolize. The last thing that you have to keep in mind when you're talking about biochemical reactions is energy. How is the energy involved? Do you have a net input of energy, or do you have a net output of energy with the reaction? Living cells can perform hundreds of different types of reactions, but all those different reactions fall into two basic types. And the first type are called anabolic reactions. And I've already given you an example of an anabolic reaction in a previous chapter. And that was dehydration synthesis. So dehydration synthesis is an example of an anabolic reaction. And with anabolic reactions, the cell is building larger molecules. And for dehydration synthesis, that's what it shows you in terms of what's happening on a molecular level. You're taking two smaller subunits and making something larger. And in order to do this, you have to break two pre-existing covalent bonds to allow these subunits to form a bond together. And when you do that, you're breaking a hydrogen off one side and OH off the other side, and then these two will come together to form a molecule of water. So that's why it's referred to as dehydration, because you end up forming or removing a molecule of water. So that's how water is involved in this reaction. The last thing to think about with this biochemical reaction is how energy is involved. And in dehydration synthesis, this is an endergonic reaction. Endergonic. And it's endergonic reaction because you have a net input of energy. So it requires more energy to cause this reaction to go forward than you get out of it. Second type of reaction are catabolic reactions. And catabolic reaction, hydrolysis is an example that I gave you before. So a hydrolytic reaction is a type of catabolic reaction, and catabolic reactions are where you are breaking things down. So in this case, you're taking a larger molecule and breaking it down into smaller parts or breaking a subunit off. And in order to, so that's what's happening on the molecular level. In order to do that, you are breaking a covalent bond to separate the subunit from the larger molecule. And in doing that, you're still going to need to form a covalent bond on the subunit and a covalent bond on the larger molecule. So water does this. So that's why this is called hydrolytic reaction, because you're splitting a molecule of water. So an OH from the water will make up this covalent bond. The hydrogen from the water will make up that covalent bond. And so it's hydrolytic. In terms of energy, these tend to be exergonic. Because there is usually a net release of energy. So that's what's happening in terms of energy. So all the reactions that you find in a cell, they can be divided into either anabolic reactions or catabolic reactions. And so remember to be able to describe these in terms of what is happening with the molecules, how water is involved, and how energy is involved. ATP stands for adenosine triphosphate. And ATP is the universal energy currency for the cell. And what that means is it's used by all cells and for all their reactions that require energy. So it's the universal, readily usable form of energy. And the structure for ATP is that it has an adenine group, which is a nitrogenous base. It has a 5-carbon sugar, a ribose, and then it has 3 phosphate groups that are bound together. So the basic structure should remind you of a nucleotide, so an adenine, nitrogenous base, a sugar, and a phosphate group. And this is classified in with the nucleic acids, so it is very similar to a nucleotide. Now the energy in the ATP molecule, the potential energy, is in this last bond. So this bond between the last phosphate group and the second to last phosphate group. So this is a very sort of unstable bond because you have this phosphate group with a negative charge that has been bound to another phosphate group with a negative charge. And so they don't like being next to each other. There's a lot of repelling. Like if you had two magnets and you took the same end and tried to push them together, they repel against each other. So there's a lot of potential energy in this bond, and when it's broken, that releases a lot of energy. And that's where the energy is in the ATP molecule. This slide shows how ATP is cycled into ADP and then cycled back to ATP, and it shows you where the energy or how energy is involved. So here's your original ATP molecule. And ATP triphosphate has 3 phosphate molecules bound to it. And remember, this last bond is highly unstable, so it has a lot of potential energy. When the cell needs to use this energy, it is going to break off that phosphate group, and that releases energy. So this is an exergonic reaction. So it's going to have a net release of energy. And it is a catalytic reaction because you are making the ATP molecule smaller. You're breaking off that phosphate. After that phosphate is broken off and energy is released, now you are left with an inorganic phosphate, the phosphate group by itself, and a DP molecule. ADP is adenosine diphosphate because it only has 2 phosphate molecules bound to it. And ADP is more stable and has lower potential energy. So you can't really use this as well for energy as you can for ATP. So what the cell will often do is recycle this ADP, and it wants to form another ATP from that. So to do that, that's going to be an endergonic reaction. So when you bind that 3rd phosphate to the ADP, now this is an anabolic reaction. So now you're building something larger, you're putting a phosphate on the molecule, and that is going to require a net input of energy. So an anabolic reaction and endergonic reaction. But that's how ATP is cycled with ADP and how the energy is involved. When ATP is broken down, you release energy. To form ATP back from ADP, it requires an input of energy. Phosphorylation is the term we give to explain how ATP is facilitating reactions. So as I mentioned before, ATP is going to release its energy by breaking off a phosphate and leaving an ADP molecule, adenosine diphosphate. And in doing so, that release of energy is going to cause reactants to be converted into products. Now down here, this is a little bit more detailed on what is actually happening. So here again, the ATP, it is being broken down to an ADP, and here is the phosphate, the inorganic phosphate. And what actually happens is that that phosphate is attached to a molecule. So originally this molecule, this protein, was inactive, it wasn't activated, and when the ATP transferred its phosphate to that molecule, it activated the molecule, and now the molecule can facilitate a reaction. And that process is called phosphorylation because this protein has been phosphorylated. It's had a phosphate group added to it. There's several different functions or different type of work that ATP can be involved in a cell. First type of work is chemical work. So ATP serves an important role to facilitate anabolic reactions. And this is an example of forming a peptide. So linking 2 amino acids together to form a peptide. So this is the first amino acid. So here's the first amino acid. And the purpose is to link it to the second amino acid. And to do this, the first amino acid needs to be phosphorylated. So here's the ATP, this is the ATP, this is the 3rd phosphate. This bond is broken. The first glycine amino acid is phosphorylated, and that changes its shape to allow it to bond to the second glycine. So now you have linked those 2 together, and now the phosphate is removed. So that is chemical work, anabolic work that ATP can be involved in by phosphorylating the monomers, which will allow them to bind together. Second type of work, this is with transport. So as I mentioned before with the cell membrane, some molecules are involved in active transport. such as protein carriers. And protein carriers, they have to change shape to transport molecules across the membrane. And that requires an energy input because the movement of molecules is against the concentration gradient. And here it's a little small, but here you have ATP, and if you can see what it's doing, it's adding a phosphate to the protein carrier. So it's phosphorylating the protein carrier. That changes the shape of the protein carrier and allows it to transport molecules, out of the cell in this case. All right, the last type of work that ATP can perform in a cell is mechanical work. And mechanical work, this can involve movement of the cilia, the flagella, the spindle fibers that separate the chromosomes during mitosis, and even muscle contraction. So this is an example of muscle contraction, and it's a little difficult to see, but here's the ATP molecule, and what it will do is phosphorylate protein fibers that are involved in contraction. And so that's what it is doing, changing the shape of the protein fiber and allowing it to contract. So for all of the work that ATP does in the cell, all of its different functions, it involves phosphorylating proteins, which changes their shape, which allows them to perform work. So far in this lecture, I've talked about energy, metabolism, and ATP, and this is a figure of a cell that relates all these concepts together. So basically with metabolism, an individual cell is going to take nutrients in, and then most of those nutrients are going to go through a catabolic process to break them down into smaller molecules. Some of those molecules will be used for anabolic reactions. Anabolic reactions involve building larger molecules, such as the macromolecules. Once the cell has built the macromolecules that it needs, it's going to use those for many different processes. Sometimes it will use them to build cellular structures like membranes and ribosomes. Other times, those macromolecules will be used for specific cellular processes like cell growth or cell division, or those macromolecules can be used to store excess energy. So those would be the carbohydrates, short-term storage, or the lipids, long-term storage. And then those molecules that are storing energy at some point, if the cell is not able to take in nutrients, those molecules will be broken down by catabolic reactions to provide energy for the cell. So ATP, how ATP is involved. ATP is often produced by catabolic reactions. And then when that ATP releases that third phosphate, that energy can be used for the anabolic reactions, the endergonic reactions. And then that ADP that was formed when ATP released its energy, it can use the energy from catabolic reactions to be cycled back into ATP. So again, ATP is used to power the anabolic reactions because they require a net input of energy, and ADP can be cycled into ATP by using the energy released from catabolic reactions. Now, in terms of energy itself and the law of thermodynamics, all of this involves energy being converted from one form to another. So the cell is not able to create its own energy, and when it uses energy, it doesn't destroy energy. But the second law that involves entropy, as you change energy from one form to another, you're going to lose some of that energy in the form of heat. And heat is increasing the entropy of the universe, basically. So as you convert energy from one form to another, you are releasing some heat, which is basically disordered energy. Because cells are very small, we tend to think that they are very simple in their function, but this is definitely not the case. This is an example of all the known biochemical reactions that can occur in an individual cell. And in this diagram, they have color-coded them by the type of macromolecule that is involved. So everything that has a teal color, that is involved in lipid metabolism. Everything with sort of this blue color is carbohydrate metabolism. The orange color has to do with amino acids and protein metabolism. And then up here, the red has to do with nucleic acid metabolism. And everything in purple has to do with energy. And so as you can see, there are many different types of biochemical reactions, and the cell has to be able to control every single one of these. Often when we talk about chemical reaction, the implication is that it takes only one reaction to convert reagents to products. But in reality, that is not true. So this is an example that is more realistic when we talk about metabolic pathways in a cell. So in this case, it takes 3 reactions to convert a starting molecule, so a starting molecule or a reactant, into a product. So in this case, you start with the starting molecule, you go through reaction 1 to convert it into B, then B is converted into C, and C uses a 3rd reaction to actually get converted into the final product. And that is a much more realistic view of what is actually going on in biochemistry. This is an example of part of the metabolic pathways of a cell, and this has to do mainly with energy production. So this green line here, this is basically following cellular respiration. So this is glycolysis, and then it leads into the citric acid cycle or the Krebs cycle. And when we go through cellular respiration, I'm going to talk about that in more detail there. But basically what this shows you is that you start with glucose, and then as it is converted into different intermediates, it then goes through the citric acid cycle where most of the energy is removed, and then that energy is used to generate ATP. So that is the main process in this diagram. Now the thing about a cell using multi-steps to convert substrates into products, this allows the cell to release energy in small increments. So it can release the energy that's in the glucose a little bit at a time. So that allows the cell a lot of control. Also, by doing this, it is able to regulate each step. So it can regulate each step to determine if this next step is going to go forward or not. And there's a lot of interaction with other pathways. So if you notice, there are a lot of other pathways that have to do with other types of reactions that are associated with this main pathway. So up here you have purine biosynthesis. Purines are a type of nucleotide. And down here you have production of fats. So there's a lot, and over here you have some amino acid production. And the thing about these pathways is that they usually can go both ways. So for instance, if a cell is breaking down too much glucose and producing more ATP than it needs, and it actually is getting ready to divide and it needs more nucleotides because it's going to copy its DNA, then it can actually divert some of these reactants, some of this glucose, to go into a pathway to produce nucleotides. Conversely, or another example, is if it's producing too much ATP, doesn't need all that ATP, it can divert some of this pathway to produce fats for long-term storage. Conversely, let's say the cell is not getting a lot of glucose and it needs more ATP, then it can take the fat that it stored or fat that it has been exposed to, and it can funnel this fat down into this process to generate ATP. Or if it was really desperate, the cell, it can start breaking down proteins into the individual amino acids, and it can use those to produce energy. So the point of having all these interrelated biochemical pathways with many different steps allows the cell to adapt and respond to the changes in the environment. An analogy that I like to think of is driving and using a map. So of course this is a map of Illinois, And let's say you want to go from Chicago, you're starting in Chicago, and you want to end up in East St. Louis. So you can think of this as a reaction. Chicago is your reactants, and you want to have the end product of you being in St. Louis. And so, of course, the direct pathway would be to go this way to end up in East St. Louis. But let's say there's a big traffic jam or construction in Springfield, and now that option isn't open to you. Does that mean you can't get to East St. Louis? No, it doesn't mean that, because you can use an alternate pathway. So you can use an alternate pathway to get to the end product that you want to get to. Or let's say some reason you start going down your pathway to get to East St. Louis, and for some reason the environment changes, conditions change, and you want to go to Indiana instead. Well, you can, because you have so many different places where you can stop and change. You can instead change your direction and go to Indiana. So like a map, like driving a car with a map with different roads that have different intersections, the biochemical pathway of the cell allows the cell to have alternate pathways if one of them is not working, or to change the end product if conditions have changed. Controlling chemical reactions is very important, especially for a cell that needs to respond to changes in the environment. And for biochemical reactions, the main thing that is occurring during a chemical reaction is a change in covalent bonding. So in this example, you have these molecules. They could be disaccharides, two monosaccharides bound together by a covalent bond. In order to have a catabolic reaction, so to break them into their monomers, you have to be able to break those covalent bonds. So a major part of controlling chemical reactions, especially for a cell, is being able to break those covalent bonds. The first thing that is required for a chemical reaction to take place is sufficient energy to destabilize the covalent bonds to allow the products to be formed. So in this example, you have the reactants. These are the two reactants, and this reaction must be occurring in a gas or a liquid because they are moving with significant kinetic energy, significant speed. And when they collide with each other, because it is high enough speed, high enough kinetic energy, that destabilizes the covalent bonds, which will allow the products to be formed. So it allows that oxygen to leave this molecule and attach to the other molecule. So the first thing that is needed, especially for an inorganic reaction, is that you need sufficient kinetic energy to destabilize those covalent bonds. Second thing is that you need those reactants to interact in a particular orientation. So here again, you have the reactants, and now they are moving with sufficient speed. So they're moving with enough kinetic energy that they would be able to facilitate a reaction, but now they are not interacting in the correct orientation. So you have that orientation, you have another orientation, but neither of them are the correct orientation. So even though they have enough speed, they will not form, that interaction will not form the products because they were in the wrong orientation. So in especially inorganic situations, you need the molecules to have enough kinetic energy. You need them to be moving at a significant enough speed, and you also need them to come in contact in the correct orientation to facilitate the reaction and produce the products. For a chemical reaction to occur, it is important that the reactants are moving with the correct amount of energy, kinetic energy, the correct speed, and also that they interact in the correct orientation. If the reactants do this, then you will have product formation. One way to increase the likelihood of a chemical reaction is to increase the concentration of the reactants. So if you increase the concentration of the reactants, you are increasing the likelihood that they will come in contact in the correct orientation and with the right amount of kinetic energy. Another way to increase the likelihood of a chemical reaction is to increase the speed of the reactants. So if they are moving faster, they are moving with more kinetic energy, then if they come in contact in the correct orientation, you should have increase in the chemical reaction. One way in inorganic chemistry to increase the speed of the reactants is to stir them. So as you stir them, you mix them around, the molecules are moving at a faster speed and you're increasing the likelihood of the reaction. Another way to increase the speed of a chemical reaction is to heat it, and that's a very common thing in inorganic chemistry. Controlling reactions in a cell is very important to the function of the cell because cells need to be able to respond very quickly to changes in the environment. So cells cannot just rely on waiting for spontaneous reactions to occur. And they cannot increase the rate of the reaction by increasing the concentration of the reactants or by increasing heat. So the way cells control their reactions is by using enzymes. Enzymes are biological catalysts, and a catalyst is something that makes a reaction more likely to occur. And these are the catalysts that cells use to control their reactions. Activation energy is the amount of energy that is needed to destabilize the bonds, the covalent bonds in the reactants in order to facilitate the formation of products. So in this case, the reactants, it's a molecule that is composed of two subunits, A and B, and in this reaction, those subunits are going to be separated from each other to form a separate A and separate B, which are the products. And in this example, the amount of energy that is required to do this is this much energy. This is the activation energy, or the energy of activation. And that, again, is the amount of energy that is needed to destabilize this covalent bond that is holding those two subunits together. So again, to separate those two subunits from each other, you have to destabilize the covalent bond so that it can break. And an enzyme, what an enzyme does is it lowers the amount of energy required to destabilize those bonds. So it's going to make this reaction more likely to go forward because it's reduced the amount of energy that has to go into it. Anabolic reactions like dehydration synthesis involve destabilizing covalent bonds. So in this case, this covalent bond and this covalent bond need to be destabilized so that then these two subunits can form a new covalent bond to each other. Hydrolytic reactions, which are a type of catabolic reaction, also involve destabilizing covalent bonds. And in this case, this covalent bond needs to be destabilized so it can break, and so this subunit can separate from the polymer. So in either case, it doesn't matter if it's an anabolic reaction or a catabolic reaction, covalent bonds need to be destabilized, and that's what the activation energy does. It destabilizes those bonds so they can break and allow to be reformed to form the products. This slide shows the basic structure or the basic anatomy of an enzyme. So the enzyme is composed of an apoenzyme. The apoenzyme is the largest part of the enzyme, and it is composed of protein. And by itself, the apoenzyme is inactive. In order to become an active enzyme, the apoenzyme needs to bind to a coenzyme or cofactor. And the difference between these two is that coenzymes are composed of organic molecules, so carbon-containing molecules, whereas cofactors are inorganic. So this would be like an ion or a metal. So together, the apoenzyme with its coenzyme or cofactor will form the holoenzyme. And the holoenzyme is what we think of as the enzyme, and this is in its active form. And the important thing to notice about the enzyme, the holoenzyme, is that it has an active site which has a particular configuration which will fit the substrate perfectly. And the substrate is the molecule that the enzyme is going to convert into a product. Another thing to notice is that this active site, which has the perfect shape for the substrate, is a combination of part of the apoenzyme and part of that coenzyme or cofactor. So by themselves, the apoenzyme does not have a complete active site. The coenzyme does not have a complete active site. But together, when you have the coenzyme or cofactor bound to the apoenzyme, then you get a functional active site. This slide shows how an enzyme catalyzes a reaction. So here is the enzyme. Here's your blue enzyme. This is the substrate, and the enzyme is going to convert the substrate into these products. And the way the enzyme functions is this is your holoenzyme, so it's a combination of the apoenzyme and a cofactor or coenzyme. It will have an active site, so it has a complete active site, and the substrate fits perfectly in the active site. So often in textbooks they say lock and key. I kind of think of like puzzle pieces. So it's going to fit perfectly. Now, when you have that enzyme substrate complex, what is going to happen is something called induced fit. An induced fit is where you get hydrogen bonds forming at that interface between the substrate and the enzyme. So here I'll draw an enzyme. So this is your enzyme. This is the active site, and the substrate is going to fit perfectly into that active site. And then what happens are these hydrogen bonds form. And actually I should do dots. So hydrogen bonds form all over that interface between the substrate and the enzyme. And all of these hydrogen bonds, remember, individual hydrogen bonds are very weak, but when you get a lot of them, you get a very strong force. And so what those hydrogen bonds are going to do is, because it's such a strong force, it's going to cause stress on the covalent bonds in the substrate. So it's going to stress those covalent bonds in the substrate to the point where they break and reform. So once those covalent bonds have broken and reformed in the substrate, now you have produced the products. So that those covalent bonds have broken there, and now these products, because they're different than the substrate, they are released, and during this product, the enzyme is unchanged. So because the enzyme is unchanged, it can go back, and if there's more substrate, it can do the same thing again. This is an example of that induced fit that I was talking about. So this is the enzyme, and enzymes are usually much larger than their substrates. So here's the substrate, and the substrate is going to fit perfectly into that groove. And actually, because of the hydrogen bonding, once the substrate goes into that groove, then it's as if the enzyme sort of curls around the substrate because of all of that hydrogen bonding. So it's as almost if the enzyme is grabbing hold tightly of the substrate. And again, that's all due to that hydrogen bonding at the interface between the substrate and the enzyme. There are several different factors that affect enzyme activity. And the first one I want to go over is substrate concentration. So this is a graph that correlates substrate concentration to enzyme activity. And it's a pretty straightforward relationship. As substrate concentration increases, the enzyme activity is going to increase very rapidly, but then it will plateau. And the reason for this is that enzymes work very quickly. So a general estimate is that they can catalyze 10 million reactions per second. So with low substrate, you have more than enough enzyme to catalyze all the reactions for the reactants that you have. As you increase the concentration, the enzymes work very quickly, so they're going to catalyze reactions very quickly. But then at some point, you are going to have so much substrate that the enzymes can't catalyze it any faster. So every enzyme has sort of a maximum rate that it can catalyze at. And once you have saturated the enzymes, they're working as fast as they can, they can't work any faster. So even though you add more and more and more substrate, you've reached a maximum rate for those enzymes. And that's the saturation rate. The other factors that have an effect on enzyme function actually have an effect on enzyme structure. So it's very important to realize that most enzymes are composed of proteins, and protein structure is very important for its function. And we often say structure equals function. And just as a review of how structure relates to enzyme function is that every enzyme has a particular shape so that it has a particular active site. And that active site only fits one particular type of substrate. So the enzyme fits one particular type of substrate and catalyzes one type of reaction. So in this case, it looks like a catalytic reaction because it looks like that substrate is converted into two smaller products. But in any case, in this case, this is the special active site, has a particular shape. If that shape changes in any way, this enzyme will no longer be able to bind that substrate, so you will no longer get that reaction. So the structure of the enzyme is very important to its function. And just as a little review of protein production and protein structure, most genes in a cell encode the information to make a protein. So the sequence of DNA is going to directly determine the sequence of the amino acids in a protein. And one way to control enzymes is to control whether they're produced or not. If the gene is not allowed to be transcribed and translated, so if the gene is not expressed, then the protein won't be produced. So that's one way to control the function of enzymes, is whether they are actually produced or not. If the protein is produced, if the enzyme is produced, then the structure of that protein, the structure of that enzyme is very important. And remember that proteins have 4 levels of structure: primary structure, secondary, tertiary, and quaternary. And the primary level of structure is just those sequence of amino acids. Secondary structure is the hydrogen bonding between components in the backbone of that polypeptide, and you either get an alpha helix or a beta pleated sheet. Tertiary structure is how that whole thing binds together, and that involves more hydrogen bonding between R groups and ionic interactions, hydrophobic interactions, disulfide bridges. And then quaternary structure is how that protein component interacts with other proteins to be a functional protein or a functional enzyme. So these are important levels of structure. If they are disrupted in any way, then the enzyme will not function. And hydrogen bonding is one of those important bonds involved in the structure of an enzyme, because it's involved in secondary structure and tertiary structure. Denaturation is the term we use when a protein or an enzyme loses its structure and is no longer functional. So this is an example of a protein in its functional form. So it has a 3-dimensional structure here. You can see alpha helices and a beta pleated sheet. And again, the important thing is that hydrogen bonding is very important to holding the structure together. If anything disrupts the hydrogen bonding, then the whole 3-dimensional structure unravels, and then you are just left with that long chain of amino acids covalently bonded together. And then, of course, because this no longer has the appropriate structure, it's denatured, it will not function. So now I'm going to go back to factors that affect enzyme activity. The first one I talked about was the substrate concentration. But the next one I'm going to talk about is temperature. And increasing temperature can denature proteins. And you know this if you have ever fried an egg. So when you look at an egg, a raw egg, the egg white, so the egg white is composed of protein. And in its natural form, it is clear and kind of viscous looking. But if you apply heat to it, if you increase the temperature, you're going to denature that protein and it's going to change its characteristics. So it's going to become opaque white and solid. And with this type of denaturation, it's high enough heat, you have permanently denatured it. There's no way to take this cooked egg white and get it back to its normal form. So that's an example of how temperature can affect proteins. For enzymes, this is a graph of how temperature affects enzyme activity. So here's temperature in Celsius increasing, and this is enzyme activity. Every enzyme has an optimal temperature where it works at its fastest rate. So this would be the highest level of enzyme activity. And often, if the enzyme is associated with a human, that optimal temperature would be body temperature, which is 37 degrees Celsius. So this could be the graph for a human enzyme. Now, if you increase temperature beyond the optimal temperature, you'll get a little bit of activity, but then you'll get a steep decrease. And the reason you have the steep decrease is because you are denaturing the protein. So just like cooking an egg, you're changing the structure of the protein, and when you change the structure of the protein or the enzyme, it no longer functions. And for enzymes, that usually means they stop functioning. So as you increase temperature, you're going to denature the enzyme and it will decrease its activity. Decreasing temperature is a little bit different. When you decrease temperature, what you are doing, you get a gradual decrease in activity, and that's because you are inhibiting induced fit. So the protein doesn't denature, it doesn't lose its three-dimensional structure, but it can no longer perform induced fit. And often, the effect of reducing temperature can be reversed. So if you take an enzyme and you cool it down, but then you warm it back up, it will become functional again. So that's different than increasing temperature. When you increase temperature, you do a permanent denaturation, and then you can't go back. You can't go back and cool the enzyme down and have it functional again. So that's the same, think about cooking the eggs. There's no way to get this back to that form again once you have permanently denatured the protein. Another factor that can affect enzyme activity is pH. Changing the pH of the environment that the enzyme is in can denature the enzyme. And you can actually use food again as an example of how changing pH can denature proteins. So an example for this is shrimp. Raw shrimp, if you've seen raw shrimp, the meat is kind of translucent and the coloring around it is kind of bluish, sort of a bluish gray. But if you take the shrimp and you expose it to lemon juice, which is an acid, then what will happen is the meat will look like it was cooked. So it will become solid white and you'll see the pink color that you normally see on cooked shrimp. And so this is just because of the acid. So no heat has been added to the shrimp, but sometimes they say the acid cooked the shrimp, but it didn't cook the shrimp, it denatured the proteins the same way heat would. So that's why it looks like it had been cooked. So this is an example of pH and how it relates to activity. So here's pH, increasing pH, and the y-axis is enzymatic activity. Every enzyme has an optimal pH. So it has an optimal pH where it functions at its highest rate. And that optimal pH is often associated with the optimal pH for the organism it's in. Now, if you increase the pH away from the optimal, you're going to get a fairly steep decrease in activity. And that's because you're denaturing the proteins. And again, once you denature them this way, there's no way to get them back to their original form. So that'll be a permanent denaturation. And interestingly, with pH, same thing happens as you decrease pH. As you decrease pH, you get the same sort of effect. And so that's why this curve is very symmetrical. So increasing pH or decreasing pH is going to denature the structure of the enzyme. And that's because changing the concentration of pH changes the ability of the enzyme to hydrogen bond, the hydrogen bonding in its structure. The last factor that can affect enzyme activity are inhibitors. So going back to this enzyme, here is the enzyme. This is your holoenzyme. Here is the active site. This is the substrate that is specific for that active site. The enzyme will bind the substrate and catalyze a reaction. First type of inhibitor is called a competitive inhibitor. And the competitive inhibitor competes with the substrate for the active site. So it's kind of like a race. If the competitive inhibitor gets there first, then it binds to the active site and now the substrate cannot bind. So that is a competitive inhibitor. A non-competitive inhibitor does not bind to the active site. Instead, it binds to the allosteric site. And allo means other, steric means form. So when the non-competitive inhibitor binds to the allosteric site, it causes a change in the shape of the enzyme. And because it causes a change in the shape of the enzyme no longer has the correct active site, and now the substrate can't bind. So that is a non-competitive inhibitor. The last topic I want to talk about in this lecture are the redox reactions. And redox reactions are actually a combination of two reactions. One is oxidation, the other is reduction. And the redox reactions involve transfer of electrons. In oxidation, oxidation is where an atom or a molecule gives away an electron. So it's the loss of an electron. So here's this blue atom or molecule. It has this electron, and during the course of the reaction, it's going to give away that electron. So it's going to be down one electron. Reduction is the gain of an electron. So in this case, this red molecule, during the course of the reaction, it's going to end up with an electron, with an extra electron. So it's reduced. So oxidation and reduction always occur together because electrons are not all just floating around by themselves in liquids or in gas. They're usually always associated with an atom or a molecule. So if something is oxidized, if something's going to lose an electron, then something else must be reduced and gain an electron. And I know it sounds backwards, but it's easy if you just remember that reduction is the opposite of what you think. It's going to gain an electron. Another example that we've already talked about of oxidation and reduction is ion formation. So remember sodium as the atom, it has only one valence electron, and so in order to become stable, it's easy for it to just give away that electron. So because it gives away an electron, it is oxidized. And chloride is going to take that extra electron because it only needs one more to have a completed valence shell. So it's going to accept, it's going to gain that electron, so it's going to be reduced. Redox reactions are very important in biology, especially in processes that involve energy like photosynthesis and cellular respiration. And redox reactions involve the transfer of electrons. Now in biology, especially biochemical reactions, we can't see the transfer of the electrons, but electrons often move or are transferred with a proton. And one electron plus one proton equals a hydrogen atom. So it equals a hydrogen atom. So one of the ways we can identify reduction and oxidation in an equation is to look at the movement of the hydrogen atoms. And all of this involves or implies a change in energy. So if something is reduced, that means it gains electrons, so it's gaining hydrogen atoms, and often it is gaining energy. If something is oxidized, that means it's losing electrons, it's losing hydrogen atoms, and it's probably losing energy. So for photosynthesis, if you look at the reactants and the products, you can determine what is oxidized and what is reduced. So in photosynthesis, the overall equation, carbon dioxide and water are the reactants. The cells or the plants use energy from light energy to convert those reactants into glucose and oxygen gas. So if you look at this, the carbon dioxide is going to gain hydrogen. So the carbon dioxide is going to gain hydrogen. So it's gaining hydrogen, it's gaining electrons, gaining energy, so it's reduced. The water is going to lose its hydrogens. So it's going to lose hydrogens, lose electrons, so it is going to be oxidized. The other important process that we will be talking about in class is cellular respiration. And cellular respiration is the process by which cells take glucose and oxygen as the reactants, and they run that through a series of reactions to release energy. And in doing so, they release the waste products, carbon dioxide and water. So glucose and oxygen are the reactants, carbon dioxide and water are the products. So during this process, glucose is going to lose its hydrogen. It will be converted into carbon dioxide. So glucose is losing its hydrogen atoms. It's losing electrons. It's losing energy. So it is being oxidized. Then conversely, oxygen, the oxygen gas, is going to gain hydrogens. So it's gaining hydrogens, gaining electrons, it's being reduced. So this will be important when we go through the two processes. We'll spend a chapter on photosynthesis and we'll spend another chapter on cellular respiration. This is the end of the lecture for chapter 6 on metabolism, energy, and enzymes. And this went over basically those concepts and how they relate to individual cells. So all living things need energy because they need to metabolize. The readily usable form of energy is ATP, and they use enzymes to control their reactions.